Hey, Kerry again with Best of Us Investors, the guy with the cool coffee cup. I've told you on a number of occasions that the channel is going through a transition, and what I want to make it is your toolbox, the toolbox you can go to make better investment decisions. And in that toolbox, we have introduced you to a platform called Stock Card. Jacob shared it with you as a AI means of searching stocks and finding stocks that you want to learn more about. We've talked to you about the trading views, the charting system I use that I call my psychiatrist to give me views into the past so I can feel more comfortable with what's in the future and make good investment decisions. Then I talked to you about Seeking Alpha, the means by which you can go learn more. I mean, one of the ways you can learn and educate yourself is through reading, but also, and that's books, but also through things like Seeking Alpha that you can gain some strength and gain some knowledge. Then we have our Discord. That's where you go to Best of Us Investors. You come join us as a group, as a tribe to make better investment decisions. Someone said I should not call it a tribe, I should call it a community. Whatever you want to call it, you call it that. Then we've added some presenters. Trent, my son, who just recently talked to you about the investment opportunity in bonds that is currently presenting itself. Mark will be coming in this next week. He is more of a value investor. And then Jacob, who you've met before, is going to talk more about bioscience. And that's what I want to do. He is bringing us an interview with the head scientist of immune sciences, and they're going to explain the drug that they're developing. And then he's going to follow up with another video with the CEO of the company. And this is some in-depth ways for you to become a better investor. And that's what I want Best of Us Investors to become. We're not just a coffee cup maker. We are, in fact, we make hats as well. We want to be your source for knowledge. Now, do we invest like other YouTube channels do? Maybe not. We're not looking for that flash in the pan. We're looking for that investment that maybe is ahead of the market but has more in-depth knowledge. What I want to do is not be your financial advisor. I want to educate you. So stick with this. Right after this, you're going to meet Jacob and the head scientist. Yes, the head scientist. So listen closely. This is deep.
Best of Us Investors presents Kerry Griegmeyer. Hello, everyone. Thank you for watching this video. We have a real special guest. We have Professor Mark Lowdell from INmune Bio here with us. He'll give a quick introduction, but before that, I just wanted to tell you that this is part of a 3-part video series. The first part is already up, and that's on just a basic background of the company. And then today we're going to talk about specifically their oncology drug, Inkmun, with Professor Lowdell. And then finally, we're going to be talking with the CEO of the company, RJ Tesi, Dr. RJ Tesi, and hopefully get a little bit more background on the company. So with that being said, Professor, would you be able to introduce yourself?
Yeah, hello, and thanks for doing this, Jacob. So I'm a professor of immunology, a cancer immunologist by training over many years, so 30 odd years of looking at immune responses to cancer. And Inkmun's come out of my research lab at University College London, where we identified a way of activating a patient's immune response against their cancer and took it into clinical trials, and then developed generation 2, which is Inkmun, and put it into INmune Bio. So I'll be pleased to talk to you about it today.
Yeah, perfect. Thank you. So I guess a good basic first question is, what is Inkmun and how does it compare to other NK immunotherapy platforms being developed in clinical trials?
That's a very good question. So Inkmun is very, very unique. In fact, so unique, we've coined a whole new word for it. So Inkmun is what we're calling a pseudokine. So the immune system works by cells and proteins in the blood interacting with each other. And immune cells make signaling molecules called cytokines, i.e. cell and movement, cytokinesis from the Greek. And cytokines are circulating in our body all the time, and many of them are used to activate or suppress the immune system. They control our immune response. Cytokines don't work singly, they work in unison. And for many years, people have tried to use cytokines to tailor an immune response or initiate an immune response to cancer. The first one was IL-2, interleukin-2, which activates T cells, but it also activates natural killer cells. And natural killer cells are in all of us. They are our primary defense against viral infection and cancer. And if you don't have natural killer cells, you don't even get born. You die in utero. So that they're very fundamental to us. And in fact, we share them with earthworms and sponges. So we know that NK cells are very, very important in immune response to cancer, and actually in immune surveillance. They go around your body looking for cancer cells and killing them. So if you get cancer, it's largely, or certainly partly, in many cases, due to failure of your NK cells. And so we were looking at how you might potentiate the NK cell response. We knew that interleukin-2 works, but it's very toxic to give to the doses that you need to sustain an NK response. There are other NK activating cytokines like IL-12, IL-15, and IL-18. And in vivo, they all work together. If you use them in vitro, i.e. outside the body in a test tube, you can activate NK cells really, really well to kill all sorts of tumor cells. But you can't give lots of cytokines to patients because it's very toxic. You get this condition called cytokine release syndrome, where the body becomes hyperinflammatory and it kills you. Inkmun provides many, many, many of the signals that IL-2, IL-12, IL-15, IL-18 does, and we're only now dissecting all of those signals. But it provides multiple signals at the same time. And in the lab, they activate NK cells as good or better than IL-2 and IL-15 and IL-18. And in the body where we've treated patients with these cells, they still give those multiple signals as if you'd given lots of cytokines. But because Inkmun only binds to NK cells, it doesn't bind to other cells of the immune system. And so therefore you don't get these side effects of cytokine release syndrome. We've given really quite high doses to at least the second patient we treated with no adverse events whatsoever. And yet we do see systemic activation of NK cells in these patients. And these NK cells are so activated that when we take them out of the patient into the blood, they kill NK-resistant tumor cells. So Inkmun it's a cell, it's a tumor cell, which is grown in as a medicine in a pharmaceutical manufacturing facility. And then it's made replication incompetent, so it can't form a tumor in the patient, and that's Inkmun. So the cell line is called INB16, and when it's made replication incompetent, it's Inkmun, and then that's when it becomes the pseudokine that activates NK cells like multiple cytokines.
All right, yeah, perfect. So that kind of touches briefly on my next question. And so that's what exactly does Inkmun do? So I think the best way to ask this question is, could you give a brief explanation? So just a quick summary. So if I'm walking on the street and someone asks, what exactly does it do? What's my brief response to them?
Yeah. So Inkmun binds to resting NK cells, which we have in our peripheral blood. And then we call it ligating. It ligates molecules on the surface of the NK cell that gives it critical signals to switch it on. That's basically all it does, but it gives multiple signals all at the same time and gets that NK cell from being at rest to being primed and ready to kill a tumor cell.
All right, perfect. So we have the background, we kind of have how it works. So my question is, what is the purpose of Inkmun? So what are some use cases that we have for it?
So there have been lots and lots and lots of papers over the years showing that patients with lots of NK cells in their tumor do better than patients with the same cancer that don't have very many tumor infiltrating NK cells. There are also papers showing that individuals with good NK function do better when they've got cancer than patients with poor NK function. And we only published a couple of years ago now in one of the blood disorders called myelodysplastic syndrome, And all of the patients who were still alive 2 years after initiating routine treatment were those who had good NK function. And those who had poor NK function all died within 2 years. So we know that if you can take, we then hypothesized that if you can take these patients with low, poor performing NK cells and make them have better performing NK cells, you could push them into that survivor group. And that's what Inkmune appears to be doing in terms of our biomarkers. It is moving these patients from low performing NK cells to high performing NK cells. And that we believe will lead to improved outcome in these patients.
All right, that's very exciting. So how certain are you at this stage that innate NK cells that have been primed by Inkmune will actually start killing tumor cells in the body in a manner that's sufficient to speak of as a effective drug therapy?
Yeah, well, that's the aim of all clinical trials, trying to find out whether your therapy works. In the laboratory, Inkmune primes NK cells from patients to kill their tumors better. So we've published a number of papers showing that. Most recently, I guess, in ovarian cancer, where we've taken NK cells from the peritoneal cavity of patients with ovarian cancer and demonstrated that these cells get switched on by Inkmune to kill ovarian tumors. And they do so by, part of that is making the NK cells stick to the tumor cell. And anyone that's got the time to go to our website should have a look at the videos there that are linked to the YouTube videos, because there are some great photo videos of or video micrographs of NK cells treated with Inkmune killing tumor cells, and they're great to watch. So what we can't say at this stage in the clinical trial development is whether we've cured any patients. What I can say is that patient 1 is clinically better than he was before he was treated. So he's completed treatment, he's completed follow-up, he's now 6 months post-treatment, and he is clinically better than he was when we initiated treatment. So in cancer, there are performance scores called ECOGs, and an ECOG of 5 is very sick, and an ECOG of 0 is normal. He had an ECOG of above 2, and his ECOG is now 0. And he's not transfusion dependent, he's not in hospital, and so we believe we've had a clinical effect. But how big that effect is and how long, we don't know, it's too early. We've treated other patients compassionately, and our second patient, the first patient we treated compassionately, had been in hospital for 6 months and was entirely transfusion dependent, couldn't go home for risk of infection. And a month after she completed treatment, she was discharged, and she's been at home ever since. And we were awaiting follow-up to see what effect we've had on her disease in her bone marrow. And the third patient, once again, had very, very high levels of disease. We've treated him, and we're waiting to see what his counts are. So at the moment, I can't say whether Inkmune is curative for these patients. It certainly put 3 patients in a better position than they were before they were treated. That's what the clinicians tell us. And we're waiting now to enroll more patients and go through the traditional formal clinical trial process.
Yeah, those are very exciting results, and that's part of why we're here talking to you, obviously. So my next question is, what is the durability of response for this therapy? Because obviously oncology drugs are very expensive, and that's a struggle for many oncology drugs.
Yeah, particularly in the field of cellular oncology drugs. So the CAR Ts that you're probably familiar with, chimeric antigen receptor T cell is a very expensive drug to make. It's a very expensive drug to supply, and it's specific to the patient. It's an autologous therapy, so it's absolutely unique to that patient. And I think the charges in the States are close on to $500,000 for a treatment. Now, Inkmune is very different. It's a immortalized cell line. It grows in large vats. We can make very large amounts of Inkmune very cost effectively. So it's not going to be when it is successful and when it is on the market, and I can say when because I believe in it, but that is a very affordable drug to deliver, and it's very easily delivered because it's shipped at -80, just like many other drugs, and it can be stored in hospital pharmacies much more easily than a CAR T cell or other NK cell therapy. So the NK cell therapies that are out there at the moment are all adoptive NK cells. They're either from healthy donors or they're from iPS cell lines. They require manufacturing outside the body and then shipping in large doses to give to patients. Inkmune is different because obviously it is this pseudokine. It's a suspension of cells, and they don't have to survive in the patient very long because they activate the patient's own NK cells. So you haven't got this persistence problem that you have with allogeneic NK therapies that are being rejected. These cells come in and they activate the patient's own NK cells. And because it's so cost effective, in the clinical trials, we're giving just 3 doses, one on day 1, one on day 8, and one on day 15. And in the patients treated, we've detected activated NK cells for many months after. In the clinical trial, we're only allowed to follow for 119 days. And still, at 119 days, our first trial subject had activated NK cells, which were memory-like NK cells able to kill cancer cells, right out at day 119 when we stopped formal follow-up. So the response seems to be sustained. The other patients, it's also been sustained, although we haven't monitored them for that long yet. They're still being followed. But because it's an inexpensive and easy drug to deliver, there's no reason why, like most chemotherapies, you can't repeatedly treat a patient. And you think about Gleevec, the most successful therapy in the treatment of chronic myeloid leukemia ever. No one expects Gleevec to cure a patient. Patients are repeatedly treated with Gleevec. And we could think about Inkmune in the same way. It doesn't have to be a treatment that in a single dose, like a CAR T, cures you. It can be a maintenance therapy because it's so low risk and it's so well tolerated that in an elderly patient population that couldn't be treated with something that was more aggressive, Inkmune appears to be safe and could be given in multiple treatments.
So if you're talking about using Inkmune as a recurring treatment, what does the safety profile look like? So
the patients so far have had 3 doses each, and the safety profile has been absolutely. In fact, when I said to the clinician treating the first patient treated in the trial, how was he? Because obviously, we hadn't treated a patient yet. I was very, very nervous, and he said, "Oh well, if boredom was a reportable event in a clinical trial, he's had grade 4 boredom." So it's really, really well tolerated. And even the second patient, who had 3 times the dose of the first patient, really well tolerated, no adverse effects whatsoever. So we're not expecting there to be a toxicity problem, but obviously we're very early days yet. We're at the lowest dose in the formal trial, and having seen so much activation, we may not even dose escalate if the next 3 patients show the same response.
So what cancers have you treated so far with this drug, and what cancers are there out there that you think could potentially work with this drug?
That's a very good question. So the patients treated so far have had blood cancers. So the first patient was treated with a disease called myelodysplastic syndrome, where the bone marrow is being taken over by a clone of cells that pushes out the clones that make normal blood cells. So you end up without enough cells to fight infection, called neutrophils. You don't have enough cells to stop your blood, that clot your blood in the event of a cut, or platelets, and you don't have enough red cells, which is what oxygenates your body. So in all cases, in that first patient, his blood counts returned to a level of normality that he didn't require transfusion support. So MDS is our lead indication. Our second indication in the pipeline is ovarian cancer. We're just having logistical problems in the technical issues of the catheter that you use to inject cells intraperitoneally. But that's purely an issue of our regulatory filings. It's nothing to do with the drug. We've got very, very good data in other diseases in the lab, in vitro. So we've got great data for killing another blood disease called myeloma, and I would love to do a trial in myeloma, and we're working on that at the moment. We've got very good data against breast cancer, including triple-negative breast cancer. So that's another area that one could go to. But I think after ovarian cancer, our next trial will probably be in renal cell carcinoma, where there's been a lot of NK cell work. and the problem has been sustaining large doses of NK cells in those patients. But we know that renal cell cancer is a great target for NK killing. And the other one we're looking at the moment, we've just received a collaborative funding with a group in Hong Kong, the Chinese University of Hong Kong, to look at nasopharyngeal cancer, which is a huge problem in Asia. It's a big problem in the US, but it's a huge problem in Asia. And we're just doing the experiments this week to confirm Inkmun's ability to activate NK cells to target nasopharyngeal cancer. So that's where we're going in the near future. But we've looked at prostate; it's another good target. So yeah, NK cells see lots of different tumor-related molecules. So it's not like a CAR T that's specific for a single antigen.
Sure. That's obviously very exciting that there's a large market out there. So with how does Inkmun deal with don't eat me signaling? So from my reading, my reading, I understand that this is a very relevant thing and that it is downregulated. Some receptors are downregulated because of Inkmun.
So don't eat me is something that tumor cells express this molecule called SIRP alpha, which interacts with CD47 on tumor-associated macrophages. Macrophages eat tumor cells, so you can avoid being eaten. That's not a mechanism that NK cells use. In fact, NK cells express SIRP alpha themselves, and they express CD47. So they stop themselves from being eaten by the tumor-associated macrophages in the tumor. So the "don't eat me" signal is not so relevant for NK cells.
Okay, obviously that's very exciting. So one thing about Inkmun, obviously, that you've talked about is it's very easy to manufacture and it can be manufactured relatively cheaply. Could you explain a little bit on where that stands in numbers? For example, what do you think a potential range of the drug price could be down the road?
Oh well, I'm not a drug pricing person. I'm a scientist, and drug pricing is a whole industry. We certainly manufacture at a price that is. I have several jobs, so I'm a part-time CSO for INmune Bio. But in order to make this function, I retain my university chair. So I'm still a professor at University College London, and I run a manufacturing facility for lots of clinical trials for advanced therapies. I think we're the busiest in Europe. So we manufacture CAR Ts, we manufacture mesenchymal stromal cells, we manufacture tumor-infiltrating lymphocytes. And I can tell you that compared to the other cell therapies that we manufacture, the actual cost of goods, because that's all I know, I don't know what one would sell a drug for, but our cost of goods for Inkmun is about 10% of what it is for all our other products that we manufacture.
Okay.
So it's a very cheap drug to make.
For sure, very exciting. Well, that's all the questions I had. So is there anything else that you wanted to touch on or mention while you're here?
So I think, and it's for your listeners, if you go to the INmune Bio website, there's a really nice video graphic little animation showing how Inkmun works in a cartoon and digitized fashion, which I've done the voiceover for. So it does explain in depth how Inkmun works. It shows some of the molecular interactions, but it's very accessible. So I would recommend going and having a look at that.
Awesome. Yeah. So thank you again for giving us your time and answering our questions and being a part of this. And with that being said, thank you for watching the episode, and I hope you have a good day. Thank you very much to everyone who's joined us. Cheers.
Okay. That's where this channel is going. That's where Best of Us Investors is going. I want to be a place you can come every day and you can find a source of knowledge and you can sort through it and say, yes, that's part of what I want to add into my toolbox to become a better investor. I don't want you to, it's not my place to say I don't want you. I want to expose you to a different way of approaching investing. Don't accept anything blindly on YouTube or any place else. Put your toolbox together. And it may cost you. It may cost you to get a stock card membership or a trading views. or Seeking Alpha or joining our Discord. You aren't going to, I guess it was, who was it that said something to me in college once? You aren't going to make money without spending some money. So you decide how far you want to go with this and what you're willing to invest into it. Invest financially as well as invest intellectually. I want to help you on both those fronts. Okay, again, Kerry Grinkmeyer, Best of Us Investors. We have a tribe. We meet on a rate every day, and then we have a conference call on Zoom every Friday where we sit around the fire and talk about what's happened last week and what's going to happen next week. Talk to you again tomorrow.